Hello, I'm Jane Davis, and it's my pleasure to talk to you today about antimicrobials in development. I lead a CF Trust-funded strategic research centre looking at Pseudomonas infection in cystic fibrosis, through which I work with a large number of other collaborators, as shown here. In terms of an outline, I thought it would be worth looking at the magnitude of the problem and addressing head-on the fact that I don't think this problem has disappeared or is likely to do so in the near future with highly effective modulators. We'll have a look at what's in the trials pipeline and in preclinical stages, but then also discuss briefly some challenges for future antimicrobial development. So this is a familiar graph to anybody working in cystic fibrosis. Lung infection shown here in 2019 from the UK CF patient registry. A large number of chronic infections shown here with a preponderance of Pseudomonas aeruginosa from adolescence onwards. And although these trends have changed slightly over the years, you can see here from US data from 93 to 2019, with an increase in staph and a relative decrease in Pseudomonas aeruginosa, there remains a huge burden of infection in the lung. And this is directly related to the severe structural lung disease, which causes loss of lung function and early mortality in cystic fibrosis. Why do I think infection will continue to remain a problem now highly effective modulators are more widespread in use? Data here from the combined efforts of the US and the UK patient registry in the few years after ivacaftor became available for people with gating mutations. And you can see both of these data sets show the same thing, that there is something of a reduction in the frequency with which chronic Pseudomonas infection gets reported in the ivacaftor cohort compared with their comparator group, but this still remains present in the vast majority of the patients in whom it was there beforehand. We know from data such as this by Katie Heisert that some of the initial impacts on bacterial burden appear then to plateau or even increase thereafter. So these influences appear, at least with ivacaftor, not to be maintained. I think we don't yet have enough data on triple therapy to know whether this will be the same for Elex-Tez-Iva, and we urgently need to continue to collect these data. Where I think the biggest unknown is those children who are fortunate enough to start highly effective modulators early in their lives. And of course, we may see something completely different with regards to delaying or preventing chronic infection in this cohort, which we really need further data for. So I've used the data from the CF Foundation trials pipeline really as a scaffold for some of this talk. We'll focus largely on avenues in the clinical space, but there are, of course, also drugs which are being developed outside the foundation, and I'll highlight some of these also. So first of all, at the top of this chart, intravenous gallium and an alternative approach further down on this is an area of interest and has been for quite some time. I'd like to acknowledge Chris Goss for kindly sending me some slides on this area. So gallium disrupts pseudomonal ion metabolism, blocking ion uptake. This inhibits bacterial DNA synthesis and catalase. It works best in low iron conditions. And one of the great advantages of this particular drug is it's already approved for another condition, hypercalcemia. This could give it a more rapid route into the market if it's effective in cystic fibrosis. There are in vitro data demonstrating efficacy against Pseudomonas, including MDR strains, stationary phase and in biofilm. And it's been shown to work in acute and chronic mouse lung and wound models. On the basis of this and other data, the IGNITE trial was launched. This recruited patients with cystic fibrosis to determine the efficacy and safety of a continuous 5-day intravenous infusion of gallium nitrate. There was a placebo control, which was saline. The primary outcome was change in baseline FEV1. And you can see here at these two early time points, day 6 and day 14 following the 5-day infusion, there was a significant difference between the treated and the placebo control group. Unfortunately, the primary outcome time point had been predetermined at day 28, by which point that difference was no longer seen. However, when looking at change from baseline in pseudomonal density, CFU per mil, you can see here that there was also a difference in those two dose groups at the day 28. More recently, the ABATE study is being launched, which is a multi-center open-label study in 6 to 10 sites in the US. 40 adults with cystic fibrosis, this time infected with Mycobacterium avium or abscessus. They will receive 2 5-day infusions of gallium nitrate at the same dose, separated by an interval. Those of us who look after patients suffering from atypical mycobacterial infections will know how devastating some of these infections can be and how difficult they are to treat conventionally. So we eagerly await the results of these studies. Bacteriophage is an area of great interest in several regions, and there are 2 studies currently at phase 2 in cystic fibrosis. One of the advantages of bacteriophage is they exhibit a fairly unique host specificity in terms of their bacterial targets, achieving bacterial lysis and killing the organisms. Most groups use cocktails to try and reduce resistance arising to a single bacteriophage. There's a very good recent review published in Pediatric Pulmonology, which talks about the 5 studies which have previously been conducted with phage in cystic fibrosis by either inhaled or intravenous or both routes, and targeting various bacteria, including Pseudomonas, Staph, Burkholderia, and Mycobacterium abscessus. And that's certainly worth a read if this is an area of interest to you. One of the groups who are at phase 2 at the moment have previously published this case report looking at the use of bacteriophage in a critically ill adult with cystic fibrosis. In the blue dots, you can see here the oxygen requirement during this very severe exacerbation, going up to 70 liters per minute. Several courses of intravenous antibiotics had been unsuccessful, and bacteriophage were initiated in the pink bar at the bottom, in line with a change in antibiotic therapy. So the conclusion from this case report was that this was safe and well tolerated. It may have contributed to this patient's improvement in clinical status. There's also been a very compelling case report from my colleagues at Great Ormond Street Hospital in London of a child with very severe Mycobacterium abscessus post-transplant, who was actually treated with engineered bacteriophages specifically designed for her organism. And you can see on these PET-CT scans here, pre versus post, quite a significant difference in terms of the hotspots of mycobacterial infection. This aligned with a really dramatic clinical improvement. We've been working with Amplify Biosciences in the past, looking at phage cocktails for Pseudomonas aeruginosa and finding a cocktail to which the vast majority of our clinical isolates were susceptible. But based on the very well described findings of variability in antimicrobial susceptibility over time, we wished to explore whether this might also be a feature and might influence how we recruited patients to clinical trials. This is just one patient in a number that we studied who had 4 separate isolates of Pseudomonas collected over a calendar year. They were confirmed on VNTR typing to be the same strain, and you can see a lot of variability in antimicrobial susceptibility there, but also quite substantial variability in plaque assay assessing phage susceptibility. So this is something that I think does require more work, and we need to be very careful that we're recruiting the right types of patients to these studies, not only to read out efficacy, but also to be ensured that the safety signal is actually meaningful. Moving on now to nitric oxide, which has been an area of interest for several years. This has anti-biofilm and possibly also mucolytic properties via a reduction in bacterial cyclic diGMP. Pilot studies and case reports demonstrate that inhaled route is well tolerated, reduces biofilm-associated Pseudomonas, and also has an impact on Burkholderia species and atypical mycobacteria. And there are currently 2 phase 2 clinical trials of inhaled nitric oxide. However, this drug is expensive and it's cumbersome for patients, so some have questioned whether there are alternative routes of delivery that could be used. Within the Pseudomonas Strategic Research Centre, I work with colleagues at Southampton University, led by Professor Jeremy Webb, and their collaborators at Wollongong in Australia. And they are developing the field of nitric oxide donor drugs. You can see the formula of one of those here. The particular attraction here is that inhalation is not required, and there is targeted delivery based on release of the nitric oxide in the presence of bacterial beta-lactamase. And these drugs have been shown to lead to Pseudomonas biofilm dispersion. You can see one example of this here from the SRC PhD student, Adele Soren, who looked at combinations of the donor drug DEA-C3B with colistin. And you can see that the combination of the two had a very significant impact on bacterial biofilm, whereas each on its own had only a partial impact. So this is a route being explored by many around the world, and I think shows quite a lot of promise for future drug development into the clinic. Cysteamine is a drug which is being developed by Nova Biotics, who kindly provided me with these data. It has multiple actions, both being an antibiotic potentiator, reducing bacterial virulence factor production, and there is already a small exploratory randomized clinical trial reporting a reduction in inflammatory mediators. In terms of the company's clinical development plans, they have both oral and inhaled plans in preparation. CARE-CF2 is a phase 2B/3 planned for the close future, looking at using NM001 as an adjunct therapy for pulmonary exacerbations. And then there's also a dry powder formulation. Posters being presented at this year's conference can be seen here. And then you can see a wide variety of different approaches at either preclinical or phase 1 stage by these companies. I'd like to just perhaps draw attention to the polyphore approach, which is a compound, mirapavidin, which attacks the bacterial outer membranes, as shown here. This is an area of interest for many. Akshay Savnis and Andy Edwards, collaborators of mine at Imperial College, have actually been using mirapavidin to try and potentiate the action of conventional antibiotics. And you can see here in the purple the impact of both colistin and mirapavidin together on bacterial growth in culture is very dramatic compared to either drug on its own. And synergy assessment shows that there is genuine synergy of these two compounds. One of the SRC other PhD students, Ronan Murphy, is also looking to target bacterial membranes with an aim to potentiate antibiotics. We're doing this in collaboration with Cycle Pharma and Aarhus University in Denmark, using a repurposed drug, latirumer acetate, which is marketed for multiple sclerosis. What can be seen here is that in overnight growth curves, unlike the concentration of tobramycin used on its own here, which is very, very limited in its efficacy, using latirumer acetate with tobramycin has a significant impact on this growth. Ronan has shown this for 11 clinical strains with significant reductions in MIC50 and MIC90 to tobramycin when GA is added, and demonstrated that this is actually achieved by breaching of the outer membrane and the cytoplasmic membranes of these bacteria, which can be seen in almost every single one of these clinical strains. More can be seen for interested readers in poster 520 in this conference. So there's a very active clinical space, and in the interest of time, I can't go through all of these approaches, but there are multiple different approaches being taken by many of these companies. I think there are challenges for future drug development in this space. However, preclinical development is complex. How do we assess what the best environment is to test a new drug? We know that in vitro bench testing often doesn't actually mimic what happens in the clinic, and that may be due to growth patterns being so different or the inhomogeneity of bacterial infection within the lungs. We also know that host cells can be involved and can change susceptibility to antimicrobial agents. And there's an increasing body of data looking at interacting pathogens in multi-organism environments which change the susceptibility of bacteria to antimicrobials. George O'Toole ran a very nice workshop recently, bringing together many of us from around the world to talk about this type of problem, and has come up with this output suggesting ways in which model systems should be designed to study these chronic polymicrobial infections. Clinical development is also particularly complex, and I think is going to become more so in the near future. We do have challenges with patient populations. Sputum's becoming something of a rare resource, and that can be an issue both for confirming eligibility for a clinical trial, but also if the efficacy readouts are based on sputum sampling. There is some concern that we may be going to experience some trial fatigue or a decreased enthusiasm amongst patient populations for non-CFTR directed trials, and we need quite carefully to try and manage this. And of course, there's going to be difficulties measuring change in outputs such as FEV1 on top of highly effective modulator therapy. We also have challenges convincing regulatory agencies of what sort of outcomes are both acceptable and practical as we move into this new era. So I'd like just to finish off by talking about a new initiative which has been launched jointly by the Medicines Discovery Catapult and Cystic Fibrosis Trust in the UK, forming a syndicate to address antimicrobial resistance in cystic fibrosis. The aim of this is to accelerate the discovery and translation of novel CF antimicrobials to the clinic. And the syndicate has succeeded already in bringing together a huge array of academic partners, but also pharma partners and representatives from Public Health England and regulatory bodies to try and actually design ways to address this issue. So this was a bit of a whistle-stop tour through this very, very large and complex environment. Thank you very much for your attention, and thank you once again to the Cystic Fibrosis Trust and all the people who donate into them for funding the work of the Strategic Research Centre. Many thanks.
Hello, my name is Dara Riva, and I'm the Director of Clinical Research Awards at the Cystic Fibrosis Foundation. Today, I'm going to talk to you about the Infection Research Initiative and the progress we've made to date. I'll also provide a brief update on the 2020 IRI Special Topic RFA we published in response to the SARS-CoV-2 pandemic. Thank you for joining me this afternoon, but I wanted to acknowledge that I am just one member representing the IRI team. Our team includes our co-lead Tiffany Burnett, Genevieve Mall, Jonathan Daniels, and our project manager, Kaylee Davis. I have no relationships to disclose. Infection has always been a very high priority to the CF Foundation. However, despite a robust research portfolio and therapeutic pipeline, we were still struggling with communicating the breadth of what we do in this research area. We announced the Infection Research Initiative at NACFC in 2018. To improve how we communicate our infection portfolio, we created the 6 areas of focus with the help of external advisors and community members. The areas of focus really serve to help us organize our portfolio into infection topic areas. Our research portfolio was really so much more than just finding new therapies. We were tackling infections from all angles, and we needed a better way to communicate that. To also aid in communicating our vast infection portfolio, we've created the IRI landing page. This really serves as the one-stop shop to find research priorities and currently funded projects. We also wanted to find a way to attract novel research from our academic researchers and attract new companies to come into the CF space. Well, I think when we announced a very large funding commitment, that really helped. We initially announced a goal to commit $100 million over the next 5 years. I'm proud to say that we are not far from reaching that goal in just less than 3 years since announcing the initiative. Finally, we needed to develop a process to systematically review our research portfolio to ensure we had the appropriate funding mechanisms and we knew which topics needed to be addressed to really prioritize the needs of our community. To that end, we have created the Iri Steering Committee. The committee is composed of both basic and clinical researchers, clinicians, CFF staff, patients, and parents with children with cystic fibrosis. Annually, the steering committee meets to evaluate the current portfolio and identify priorities for the coming year. They may also recommend RFA topics or topics that may require a meeting or workshop for further discussion to really drill down those research needs. As I mentioned previously, the large funding announcement we made at the kickoff of the initiative was aimed to attract new research. I do believe this was successful. Our research portfolio has almost doubled funding from 2018, and we have significantly increased not only our company interactions, but also the funding in industry projects. The 2020 IRI Special Topic RFA awarded 16 infection awards, resulting in a total of $13.2 million of committed funds. These projects were required to address one of the topics prioritized by the steering committee. Currently, we have over 200 active academic projects in our research portfolio. I should mention, though, some of those did start prior to the IRI launch. This graphic is a snapshot of our industry portfolio. Details regarding many of the compounds listed here can be found on the CFF therapeutic development pipeline. This snapshot allows us to quickly view what compounds we have and in which phase of development, as well as the target species or topic. Using the color-coded key below, you can see our funding is heavily weighted in Pseudomonas and NTM anti-infectives. However, we are actively seeking opportunities in fungal and MRSA, among other difficult to treat organisms. We also have a category for multi-pathogen targets. These compounds could either be species agnostic or work on two or more different pathogens. One of the reasons we have such a successful pipeline is due to our openness to interact with industry and provide them guidance and non-funding resources. For instance, when a company reaches out about interest to apply to the Therapeutics Development Award, or may have an anti-infective that may be applicable to our CF community, they are provided what is called an antibiotic drug development guidance document. This document is created by CFF staff and key opinion leaders in the field. The document provides guidance on things like how to conduct susceptibility testing and other preclinical work that's very relevant to the CF population. It also provides them with names and contact information to several CF antimicrobial experts. Many of these experts not only provide their expertise, but also share valuable clinical isolates that have a wide range of phenotypes and of the most common species seen in the CF population. The guidance document provides a list of unmet needs, and it also provides a list of species that are particularly difficult to treat. And lastly, the document provides a list of relevant articles to help them understand the complexity of the CF infection. We also direct companies to the Infection Research Initiative white paper that was created by the steering committee. This white paper emphasizes the importance of support on the development of novel approaches and the support of products that can treat multiple organisms. As you can see, we worked to add 3 new compounds in less than 2 years that have the ability to target multiple organisms. As I mentioned before, the IRI areas of focus serve as a method to organize the portfolio. We're able to look at each area of focus and determine if we need to adjust funding to meet the needs or fill research gaps. We tackle these gaps and needs by special topic RFAs, or if we really need to drive the research, then we may hold a workshop or meeting. In 2020, the steering committee recommended we prioritize the area improving detection and diagnosis. As we enter the new reality of more patients with CF not able to spontaneously expectorate. That year, the special topic RFA resulted in 2 new detection projects, one using urine to detect fungus and one using urine to detect NTM. However, we recognize that the challenges with detection and diagnosis required a deeper dive to understand what current methods beyond sputum are available now and what methods need to be developed for the future. The CFF has developed 2 work streams to help us tackle this important challenge: Certainly a very good problem to have. The first work stream to address detection and diagnosis was really born from the challenges caused by the pandemic. In the early months of the pandemic, many institutions paused research and routine in-person clinic visits. This, combined with the rollout of Trikafta, caused a lack of ability to monitor chronic infections, conduct surveillance, or measure response to therapy. Many care centers and research study teams were quick to adopt remote collection of sputum, including the PROMISE study, which quickly pivoted to make these important protocol changes. However, there are a lot of questions around the validity and reliability of remotely collected samples. There's also a lot of questions about how the specimen is handled and processed depending on the types of assays being conducted. The Remote Endpoints Task Force was convened with the goal to determine the accuracy, standardization, and optimal frequency of home measures. They also are going to tackle the best way to do virtual sample collection. The task force will tackle several types of endpoints, including microbiologic. We recently held an open ask for volunteers to join the task force, and I hope to begin our work very soon. As the remote collection kicks off with the task force to address some of our immediate challenges with collecting sputum, we are also keeping our eye on the future as we anticipate a generation of people who begin highly effective modulator therapy very early in life and don't have the ability to produce sputum on a regular basis. The Detection and Diagnosis Workshop is focused on developing recommendations to address the future needs, which may include a special topic RFA or more detailed priorities document related to novel approaches. This workshop is being led by Drs. Lindsay Caverly and Luke Hoffman and will occur later this month. The workshop is being divided into 3 sessions. The first really focused on defining why and how we use sputum in both the care and research setting. Second, we will review the current methods we have available, like BAL and OP swabs, and what needs they may fill in the absence of sputum. Finally, the workshop will look at novel detection methods and what research is needed to continue their development. This table shows funding commitment for the academic research from 2018 to 2020 by microorganism. Fungal was easily identified as a major gap in research at our first steering committee meeting. In fact, it was really the only microorganism specifically called out in that 2020 IRI special topic RFA. We did receive some proposals in this area, including the urine detection study I mentioned earlier, but we really wanted to drive this research a bit more. Earlier this year, we held a series of fungal workshops with the goal to design a large clinical study that will identify and characterize the health impact of Aspergillus infection and potentially set up a biorepository of specimens and clinical data to help advance novel research. Pilot work on remote collection of sputum sent to a centralized lab has just begun. This work is being led by Drs. Gina Hong and Dave Nichols. Once the pilot work is complete, study design of the full protocol will begin. We hope to share more on the progress of the fungal work by the second quarter of 2022. Bacteriophage became an area priority as we started to learn more about the positive reports of this experimental therapy for people with CF. Ella Balasa, who is also on our steering committee, has been a very vocal advocate for this new therapy. While the positive case reports were promising, we need robust clinical studies to truly understand the benefits of this therapy. We now have an investigator-initiated study looking at phage therapy in people with multi-drug resistant Pseudomonas. And as I showed on the industry slide, we also have an industry award to Armada to further develop their phage product in Pseudomonas. The large funding done in 2020 in the viral category was due to the COVID RFA. In response to the COVID pandemic and the limited amount of scientific data on the impacts of the virus on people with CF, CFF decided to launch the SARS-CoV-2 and cystic fibrosis request for applications in June 2020. This RFA was intended to solicit basic science research projects to help us better understand any biological differences in response to acquisition and propagation of the virus in CF. This work was led by Dr. Catherine Tuggle at the CF Foundation. We received 53 letters of intent and invited 31 to submit full applications. We received 29 full applications and funded 11 projects by the end of 2020. This was a one-time RFA, but investigators who are interested in submitting any SARS-CoV-2 related projects are suggested to apply to either our basic or clinical research programs. The CFF basic and clinical research programs run regular cycles throughout the year. All open funded opportunities can be found on our grants management system. The call for proposals from this RFA resulted in $2.7 million in funding for basic research to help with our understanding of this virus on a cellular level. Many of these projects started earlier this year. We have been able to accomplish so much so far under the Infection Research Initiative, but we continue to look towards the future to ensure we meet the needs of the CF community and their fight against infections. Later this year, the third steering committee meeting will take place virtually. We expect to work with our steering committee to address several key issues we see on the horizon as we embark in the new CF world. This new CF world of reduced sputum and even a healthier population with less exacerbations. We also anticipate we'll be meeting that $100 million mark. This prompted us to solicit feedback from the CF community, including researchers, clinicians, care team members, patients, and families, to help us shape the future of the IRI. Results from the survey will be presented at the steering committee meeting in December by our community members, who also helped create the survey. After the meeting, you can expect to see an update to the IRI white paper and website to keep you informed on what's next for the IRI. Well, that's it for me today. I want to thank you for listening. For more information, please visit the IRI webpage where you can find all things IRI. To see our current funding opportunities, please visit our grants management system. And as always, feel free to contact me if you have any further questions. Thank you.
Hello, everybody. Thank you very much for attending, and thank you to the organizers and the CF Foundation for inviting me to speak today on behalf of a large number of very talented people. I do have one disclosure. My institution receives compensation for the role that I play that I'm going to discuss today. These funds do not go directly to me, and our services are not for profit. So today I'm going to start with a little rationale by discussing CF respiratory microbiology. And then I will talk in general about the CF Foundation supported national resource centers and focus in on the microbiology-focused national resource centers. Starting with CF respiratory microbiology. Here is a slide I suspect everybody has seen many, many times. This is a depiction of CF respiratory microbiology in the United States for 1 year. This is from the CF Patient Registry Report for the year 2019. It's the most recently published. And it shows you the proportion of patients on the y-axis who were culture positive for all of these traditional pathogens as broken down by age. And you can see that there is some dynamics here, that this ecology tends to change as patients get older. And I also pointed out 4 organisms that I'm going to talk about a lot. The 2 most commonly cultured organisms are Staphylococcus aureus, Pseudomonas aeruginosa, and the subtype of Staph aureus, MRSA. And here's Burkholderia cepacia complex, which I'll talk about as well. This is a slightly different depiction of CF respiratory ecology. Instead of being broken down by age, this is for the entire CF population in the United States over the last couple of decades, showing you how the ecology has changed in the CF population. You can see, for example, that Staph aureus prevalence or detection has gone up over the past couple of decades, together with MRSA, and that of Pseudomonas has gone down, just as an example of how this microbiology changes as detected by culture. And while the patient registry report did not have a similar kind of graph of changing ecology in non-tuberculous mycobacteria, here's a graph that does show you just how common this emerging group of concerning pathogens are. This is a graph that shows you how many patients in 2019 with CF were culture positive for these different non-tuberculous mycobacteria. And increasingly, we are understanding that our culture methods are not perfect. They often miss organisms that we can find with DNA-based methods, such as shown in this paper from Kirk Harris, who I'll talk about in a bit, in 2007, where they compared the results of species-specific PCRs for these traditional pathogens of sputum samples. And that's in gray here, as well as black. And in white is culture only. And black is where they agree. And gray is where PCR was positive only. So you can see that they don't always agree. And increasingly, we are using not just PCR, but also sequencing-based methods, often called microbiota or microbiome-based methods, to profile the microbes that you can detect in respiratory samples. This is from sputum samples. This is one figure from a recent paper from my own laboratory. These are actually aggregate average microbiota, sputum microbiota data from 30 people with CF and their 30 sputum samples. And these samples were culture positive for just Pseudomonas aeruginosa, but you can see that sequencing picked up a lot of organisms that really weren't identified by culture. So increasingly, there's a lot of interest in these potentially very powerful methods. Now, a very important aside, there are a few respiratory sample types that we get from our patients. From young children, we generally use swabs. We get sputum and bronchoalveolar lavage fluid in the laboratory. And our preferred sample is really sputum because it nicely balances convenience and lack of invasiveness, but also a reasonably accurate depiction, we think, of lower respiratory tract microbiology, which is what most people really want to see. But increasingly, sputum is not available from our patients. It's rarely available from children under 6, and fewer children are expectorating, although researchers have shown us that inhaling hypertonic saline can sometimes induce the production of sputum. That increases the yield a bit. Similarly, adults are not expectorating as much as they used to either, and this is, of course, a very good thing, particularly with the advent of CFTR modulators, but it impacts our ability to sample these patients. And bronchoscopy, while interesting, is just not routinely done in a lot of places. There are some centers that do it often, but most of us just don't routinely for surveillance. And this issue of how we sample the respiratory tracts of our patients has very important implications for how we move forward, how we diagnose these infections going forward. And this issue will be in the background of everything I talk about today. Okay, with that background, rationale, I want to start by talking about the Cystic Fibrosis Foundation National Resource Centers in aggregate, not just the microbiology ones. They were created in large part to help the Cystic Fibrosis Therapeutics Development Network, the TDN, which is a large collaborative organization comprising a lot of specialists in CF research, including representing very diverse expertise and diverse institutions around the United States. It was founded in 1998, and since then, it has overseen or helped with the conduct of more than 150 clinical studies of a very diverse candidate therapies, such as not just anti-infectives, which is what my laboratory mostly serves, but many other types of CF therapeutics. The National Resource Centers then are funded by the CF Foundation and provide services to studies that the TDN helps with or oversees. The NRCs were established at the inception of the TDN. They are funded by the CF Foundation. And the PIs of these cores comprise what's called the National Resource Center Working Group, NRCWC. These are laboratories that provide services for TDN studies. And the NRC supports studies focused on CF with our expertise. We provide consultation. We provide resources, including laboratory resources. And we also provide both protocols and the actual services for standardized laboratory methodology. As of 2019, there were 7 national centers. This is the sole NRC in 2019 that was focused on clinical microbiology. You can see that, again, there's diverse expertise. And in general, most of these laboratories were or are embedded in busy working clinical laboratories, which, reflects the expertise that they have. However, beginning in about 2019, the CF Foundation wisely realized, as reflected on this webpage, current webpage for the NRCs, that While clinical service corps, such as those embedded in clinical laboratories, provide best practices clinical measures, in a lot of cases, we'd like to create or use or apply methods that are not available to clinical laboratories because they're non-standard. And for that, for innovating, the CF Foundation created a group of corps called the Outcome Measure Advancement Corps. And so currently, as of now, there are many more national resource centers. These are the core sites, meaning they provide what are considered to be validated or standard methods. And I'm showing you with the red asterisks here, the corps that are dedicated to microbiology, in addition to 4 new advancement or innovation corps, 2 of which, again, are dedicated to microbiology. So I'll focus on these microbiology corps first. The microbiology-focused national resource centers were first represented by one of them, called the Center for CF Microbiology. It was founded in its current form at the inception of the TDN in 1998. And when by Jane Burns, when Dr. Burns retired in 2016, sadly for all of us, she turned over the reins to me. The CCFM, or Center for CF Microbiology, has always provided best practices standard culture-based CF microbiological services embedded in a busy CF clinical laboratory at Seattle Children's Hospital in Washington. And it maintains all of the necessary, desirable certifications and approvals. It has, since its inception, provided standard CF clinical microbiological testing now for dozens of studies, including not just academics, but the studies supported by the CF Foundation itself, industry, NIH, and the FDA. Some of the early studies before the NRC was actually established, but using the same infrastructure and personnel, helped with the development of Dornase alpha, as well as inhaled tobramycin. And since then, it has supported dozens of clinical trials and observational studies, and we're currently providing services for 7 ongoing studies. The technical director, a very talented scientist named Anne-Marie Boucat, without whom I could get nothing done. However, this NRC does also provide some expertise and resources that are not commonly available from CF clinical labs, and that includes consultation. Some researchers would prefer to work with their own other laboratory or contract laboratory, and we do provide consultation and even archived protocols to make sure that these laboratories are using the best available CF or standard CF clinical microbiological testing. We also provide Staph aureus small colony variant detection and characterization, which is not uniformly available from all CF laboratories, but we have a long-standing history of doing so. In addition, there are a number of services and resources that were increasingly requested of the CCFM, but that couldn't be provided by a clinical lab. And that includes clinical isolates of pathogens such as Pseudomonas, in vitro susceptibility testing of non-FDA approved agents, candidate new therapeutics against those isolates. And clinical laboratories are precluded from testing anything that's not FDA approved. Biofilm testing or other in vitro susceptibility testing conditions such as sputum binding or sputum inhibition. And also DNA extraction for microbiome or sequencing-based analyses or other sputum or specimen processing. And because these are non-standard methods and outcomes, they require a basic science laboratory. It couldn't be performed in the clinical laboratory. And that was the rationale for creating what we call the CF Microbiological Outcomes Advancement Corps, or the MOAC. The MOAC was funded, thankfully, and began services in 2020. And it runs in parallel to the CCFM, also in Seattle, but at the University of Washington. And its goals are to create new tests and new culture methods on demand, as requested by investigators, to provide clinical isolates of standard pathogens, which we keep in an archive, to perform new or otherwise non-standard tests, such as susceptibility testing of novel agents, PCR-based detection or even sequencing of bacterial isolates from clinical trials, biofilm testing, And while we have provided sputum inactivation tests, that is the ability of sputum to inactivate or inhibit the efficacy of a novel therapeutic, this has become more limited for all the happy reasons I listed before. Increasingly, we're being asked to titer, detect, and also test susceptibilities of bacteriophage. The the MOAC is also directed by myself, but again, at a slightly different institution in Seattle, the University of Washington. And while I direct it, it's really 3 other people who do the magic, who get all the work done. And that comprises 3 subcores: the sequencing subcore led by Stephen Salaponte, the isolate and testing subcore led by Rafael Lopez, and the innovation subcore that creates new and validates new methods that's led by Dan Walter and Hilary Hayden. And we are very fortunate to have a core administrator, Donna Prybell, who keeps the boat afloat. And the CCFM and the MOAC act closely with each other in both directions. For example, the CCFM's cultures yield all of the microbial isolates that go into our archive and that we provide to people and that we test in the MOAC. We also work closely together to design, validate, and implement new tests for introduction into the clinical workflow, such as, for example, new PCR tests. And quite often in the MOAC, when we culture samples, we will identify organisms we can't speciate, and we will ask the clinical laboratory to use their very powerful methods, such as MALDI-TOF, to speciate them. So it's really a 2-directional relationship. There are a number of other microbiology-focused laboratories that are supported by the CF Foundation, and one of them is it was created and operates outside of the NRC umbrella because it's been around for so long. And this is the Burkhold Area Cepacia Research Laboratory and Repository run by John LaPuma at the University of Michigan. Again, it has been around for quite a while, and many of you probably work with them. There are also 2 NTM, non-tuberculous mycobacteria focused cores, an outcomes advancement core and a clinical core run by Jerry Nick and National Jewish Hospital. And there is also a newly created microbiome analysis core led by Kirk Harris at the University of Colorado, also in Denver. And so I'll give you a little bit of information about each of these. The Burkhold Area Cepacia Research Laboratory and Repository at the University of Michigan, again, it's been around for many years. It provides and has long provided rapid and accurate identification of bacteria that are uncommon, infecting CF airways, and they will do that on request. And that includes, but isn't limited to just to Burkhold area species and Achromatobacter species, but these relatively challenging and uncommon gram-negative bacteria are their specialty. They genotype these bacteria, and they provide active surveillance through molecular epidemiology for interpatient spread and outbreaks. And they also provide these isolates through a strain repository. And these isolates are very well characterized. And they also maintain CAP and CLIA certification. The NTM cores, again, a clinical core and its companion outcome advancement core, which is very similar to the structure of our MOAC and CCFM, is run by Jerry Nick and National Jewish Hospital, National Jewish Health in Denver. And they provide consultation, training, and services for preclinical and clinical studies of non-tuberculous mycobacteria. They maintain an archive, a large archive of well-characterized isolates from both CF and non-CF patients. And they also maintain a panel of in vitro susceptibility and animal testing models for non-tuberculous mycobacteria. They also, like the Burkhold area laboratory, provide molecular epidemiological analyses through phylogenetic analysis of clinical isolates to look for outbreaks and interpatient spread. And the microbiome analysis core, run by Dr. Harris at the University of Colorado with colleagues, provides training, reagents, and data collection for complex microbiome analyses, including study design, advice, and by a high level of biostatistical expertise for multidimensional data sets. They can define microbial community composition. They can quantify the abundance of specific organisms by quantitative PCR. And again, they collect very complex meta-omics data and provide a high level of expertise for analysis. So this slide has all 6 of those cores on a single slide just to outline or to underscore that together we provide a very comprehensive set of services for all of the organisms we think are important in CF, and also to underscore that we work very closely together to make sure that happens. For example, just using the graphs, the slides that I gave you before, together the Burkholderia core and the 2 cores here in Seattle, the CCFM and the MOAC, cover all of the services necessary for standard pathogens. The NTM cores, of course, address those emerging pathogens. And the microbiome core covers everything else. So currently, the microcores work very closely with each other, but we also work very closely with all of the other cores, including both the advancement cores and the standard cores, to make sure we meet the needs of all kinds of research. And there's some very complex research studies ongoing where we interact very closely with each other and pass data and samples and expertise onto each other. So in summary, the CF Foundation supports a number of cores dedicated to CF respiratory microbiology that includes both standard clinical cores and non-standard cores or outcomes advancement cores. And we work very closely together to provide a wealth of expertise, resources, and services. For our microcores, we're really supporting researchers investigating the microbiological determinants of disease. But we also work with all of the other cores to support CF therapeutic development in general and CF research. And all of us are also engaged in ongoing research of our own into the causes, consequences, and treatment of CF diseases, which is really the reason for these cores in the first place. So with that, I'd like to thank all of you for coming. I very much want to thank the NRC Working Group for helping me with this, these kind individuals for helping me put together the slide deck, the staff and leadership of all of our NRCs, but particularly my own, and of course, our patients and families and the Foundation for supporting us. And with that, I'd very much like to thank you.
Hello, my name is Lisa Seaman. I'm from Columbia University, and I'd like to thank the organizers for giving me this opportunity to present viral infections in CF, what we are learning during the COVID-19 pandemic. I have no relationships to disclose related to this presentation. Today's learning objectives are to describe the changed epidemiology of respiratory viruses during the COVID-19 pandemic, to discuss the possible impact of these changes on exacerbations of respiratory tract comorbidities, and to consider the potential implication of these changes on people with CF. So the impact of respiratory viruses in people with CF is well known to this audience, so I'll briefly summarize. In children with CF, respiratory viruses have been identified in 23 to 64% of exacerbations, and in 13 to 53% during periods of clinical stability. Respiratory viral infections are associated with increased cough, congestion, and sputum production, as well as wheezing. And illness may persist longer than in children without CF. In adults with CF, respiratory viruses are identified in 40% of exacerbations, 24% of periods of clinical stability, and have been associated with increased antibiotic use, higher respiratory symptom scores, and a decline in FEV1. The next series of slides will describe changes in the epidemiology of respiratory viruses associated with the COVID-19 pandemic. And the data are derived from the US World Health Organization Collaborating Laboratory System for the influenza data, and from CDC's National Respiratory and Enterovirus Surveillance System for the other viruses. I'll be sharing data from other countries as well. The first slide, I think, is potentially the most dramatic, shows us the epidemiology of influenza in the United States. And so for this and many of the subsequent slides, The y-axis will be the percentage of positive specimens, and the x-axis will be time. And you'll note that the various dotted lines and solid lines show you the previous epidemiology of influenza viral seasons, 26 to 17, 17 to 18, etc. And then the blue line is 2020 to 2021. And you may not be able to see it because it's totally imposed on the x-axis. So basically, we've had no influenza activity. And there is a growing concern that this absence of influenza might actually adversely affect the severity of the upcoming influenza season, given the prolonged absence of ongoing natural exposure to influenza viruses, which leads to lower levels of population immunity, particularly among younger children. This could portend more widespread disease and potentially more severe epidemic when influenza circulation does resume. This is a study of the epidemiology of influenza in Australia, and we'll remind ourselves that the Southern Hemisphere helps us predict what we should put into our vaccines for the subsequent Northern Hemisphere's winter. So from April 2020 through September 2021, Australia has noted historically low numbers of laboratory-confirmed influenza, community influenza-like illness activity, and hospitalizations. There was a look at the virology from 2021, and 68% of the cases were caused by influenza A. 20 samples, and that's a very small number, have been assessed to date by the WHO to look for the vaccine match. One influenza B and one influenza A H1N1 were considered to be antigenically similar to the vaccine compound. But in contrast, among the 18 influenza A H3N2 isolates, those were considered actually antigenically dissimilar. The numbers of samples and the numbers of cases were too low to calculate vaccine effectiveness. And you'll note in the figure on the right that the previous influenza seasons are shown by the dotted parts of the figure, and then that red line at the bottom is the influenza activity in 2021, mirroring our experience in the United States. The epidemiology of RSV has also been very interesting and surprising. You'll note the past years of RSV, and of course in the United States, we see RSV during the winter months, and in the 2021 season, we saw almost no RSV, and then suddenly started to see much more RSV starting in the spring and persisting into the summer. This led the CDC to put out a health advisory alerting clinicians about the increased atypical activity of RSV, initially noted in the southern US, but similar patterns have been noted throughout the United States since. This is a very important study describing the epidemiology of RSV in Australia, and it's a relatively complicated slide, so I'm going to take my time and walk you through it. So this shows us the epidemiology in 3 states in Australia: New South Wales, Western Australia, and Victoria. And the dotted red line shows us the past 3 seasons and the percentage of positive specimens. And then the solid red line shows us the RSV activity during the pandemic. And you'll immediately note that the solid red line exceeds, in the terms of the percent positivity, any of the previous epidemiology of RSV that has been noted. Another important part of this figure are what was going on in terms of interventions to slow the spread of COVID. And so you'll see for each of the figures, it might be a little hard to detect, but there is a line across the top. Wherever it's blue, that is time where either there was school closures or when there was a school holiday. So during those periods of blue, you'll see that there's very little RSV activity during the pandemic. Now, those horizontal dark gray lines are shared times when there were various interventions either removed or put into place. So, for example, in the top bar, where we're looking at New South Wales, as schools started to open and bars and restaurants and clubs started to open, you can start to see a gradual increase in the number of cases. Then, so alarmed by this wave of SARS-CoV-2, they started closing those things, and you'll see a subsequent decline. And so the authors concluded that with each opening of society, there was an increase in concomitant RSV during that time. The epidemiology of Rhino enterovirus, and we'll remember that the multiplex platforms used throughout the United States can't distinguish between Rhino and Entero, so they're lumped together, actually shows a different pattern. While in the fall and early winter, it looked a little bit lower than previous years, it really does look like Rhino Entero are returning to pre-pandemic circulation levels. Now, this epidemiology is not fully understood, and it may have something to do with different transmission mechanisms for Rhino Entero, the role of asymptomatic transition. But I also think it's interesting to consider that these are non-envelope viruses, and they actually survive for prolonged periods of time on horizontal surfaces and may not be as amenable to some of the efforts regarding surface cleaning. And then 3 other interesting patterns to share with you. The panel on the upper left are the common human coronaviruses, 229E, NL63, OC43, and HKU1. And you'll see that there was almost none of the activity, and then an increasing number of cases as the spring and summer began. And that does seem to parallel the lower left, which is para-influenza. And there's a consideration that as schools opened, we started to see circulation of both of those viruses, which tend to be more common in children. It's not a completely satisfying explanation because the panel on the right shows us human metapneumovirus, which also is very common in children. And that's that blue bar at the bottom that basically overlies the x-axis. So now let's turn to the potential impact of these changes in epidemiology of respiratory viruses on exacerbations of respiratory tract comorbidities that are thought to be prompted by infection with respiratory viruses. So first I'll share with you a study done of asthma exacerbations in England. And the top part of the panel is exacerbations that were identified in primary care. And the bottom part are exacerbations requiring a hospital visit and or hospitalization. So overall, in terms of the exacerbations managed in primary care, there was about a 50% reduction. That's that pink line at the bottom. And this was noted really with the lockdowns that occurred in the UK beginning in March. This decrease was statistically significant in all age groups, men and women, and in almost all regions in England. However, there was not a similar decline in exacerbations requiring a hospital visit or hospitalization. You note that the pink dotted line is really superimposed on the epidemiology of previous years. There was a similar study performed in Scotland and Wales, where reductions in asthma exacerbations was similarly noted. But there was, in those studies, they were noted a reduction in hospital admissions without an increase in asthma deaths. So really pointing to some interesting epidemiology for asthma exacerbations. This is a study looking at reduced COPD hospital admissions in the United States associated with the onset of the COVID pandemic interventions. So weekly COPD admissions decreased by 53% when compared to the pre-COVID seasonality. And they compared the decline in COPD exacerbations that are thought to be prompted by viral infections often with declines in congestive heart failure, diabetes, and myocardial infarction, which is not nearly as dramatic as the declines noted for COPD. There was a recent meta-analysis and systematic review that I've cited below that found a 50% reduction overall in admissions for COPD during the COVID-19 pandemic compared to pre-pandemic times, likely associated, claim the authors, with a reduction in respiratory viral infections that can trigger exacerbations. What was really important, I think, about the observations of these authors was that they say that future guidelines for COPD should consider recommendations that help control respiratory viral infections that would persist beyond the pandemic. And we'll be talking a little bit more about that in a moment. And this is a graph that I suspect is very familiar to the audience, showing a reduction in hospitalizations for CF exacerbations in the United States that was derived from the CF Foundation's patient registry. So to orient you, the orange parts of the line are individuals with CF who were 12 years of age and older, and the blue parts of the line are those people with CF who are 11 years of age and younger. And they are graphing intravenous treated pulmonary exacerbations pre and post the introduction of Trikafta, which is noted by the dotted line in the middle of the graph. And you'll see that there's a clear decline in adults who were getting the Trikafta, people 12 years of age and older, and that the intravenous treated pulmonary exacerbations in the younger age group did not decline. But then the second lower dotted line is the onset of the COVID-19 pandemic, and there does again seem to be a reduction in exacerbations requiring hospitalizations, intravenous antibiotics, that is more noted in both groups. So again, provocative. There are clearly some limitations to these types of studies. We know that there were changes in healthcare seeking behavior as people were afraid to seek healthcare during the height of the pandemic. We also know that there were changes in the availability of healthcare, and potentially the dominance of COVID-19 may have impacted diagnostic coding for exacerbations of comorbidity, particularly when both SARS-CoV-2 viral infection and an exacerbation were present in the same individual. Nonetheless, common respiratory viruses can be reduced by non-pharmacologic interventions that were implemented during the COVID-19 pandemic. And this is so familiar to all of us, but mask use, hand hygiene, surface cleaning, staying at home, and in some nations, shielding of high-risk patients who were urged to really lock down at home, school closures, physical distancing, limitations on large gatherings, reduced global travel. And there are additional benefits to people with comorbidities, improved air quality, and potentially improved self-management. So what are the implications of this for people with CF? Well, first, there's enormous value in multi-pathogen testing to best understand the epidemiology of viruses. Without that, we wouldn't have noted these off-season increases in RSV, for example. Influenza vaccination is more important than ever. It's particularly important as schools and work resume in person. All vaccinations in the US will be quadrivalent. One influenza A H1N1 and one influenza A H3N2, as well as 2 influenza Bs. But as I alluded to earlier, we really cannot predict the severity of the 21-22 influenza season. And there is concern that this may be a particularly severe season and something that we'll have to be really alert to. And clearly, vaccinations can decrease the stress on the healthcare system. that if people are vaccinated, we will reduce severe influenza and hospitalizations. And we all recognize how important that is. For RSV, there's concern that older infants and toddlers may be at increased risk of severe disease due to lack of exposure during their first year of life and sort of a lack or decrease in cohort immunity. Many jurisdictions provided off-season synergist coverage, and there is a potential for insurance issues as children would exceed their usual allocation through synergists. So how can we sort of think about this in terms of our prevention strategies moving forward for respiratory viral infections in people with CF? I want to say to the audience that we should consider continuing selected NPIs in the community even after the COVID-19 pandemic wanes, avoiding large gatherings when feasible, continued masks in school, in crowded public indoor settings, and of course, hand hygiene. And maybe in an era of personalized medicine, we could consider implementing this in selected people with CF. Obviously, individuals following a transplant, those with severe lung disease, those who are not yet receiving a highly effective CFTR modulator, or those who are unvaccinated for SARS-CoV-2. So in summary, there have been a decreased circulation of many viruses during the pandemic, most notably influenza, and an increased circulation, sometimes off-season, of some respiratory viruses, most notably RSV. Non-pharmaceutical interventions for COVID-19 do reduce the transmission of common respiratory viruses. And so in addition to current infection prevention and control strategies for people with CF used in healthcare settings, we should consider the use of NPIs in community settings, particularly for individuals who are at high risk of severe viral infections and potential exacerbations. So I want to thank you all so much. Please stay safe and healthy. Don't forget to take care of yourself. And I can't resist asking all of you to consider when the last time you had an upper respiratory tract infection since the pandemic began. So I look forward to the question and answer period. Thank you.
Okay. Are we on? Are we good to go? Okay, great. So thanks everybody for joining today. And thank you very much to our speakers. I think those talks were outstanding. So I really appreciate it. I'm Iri Steering Committee. I'm from the University of Colorado. And I just want to say hello to our speakers and to my co-moderator, Ranjani Somiyaji from the University of Calgary, and then to Jane Davies from the Imperial College in London, Dara Riva from CF Foundation, Luke Hoffman from the University of Washington, and Lisa Seaman from Columbia University. So thank you all for being here. And thank you to my co-moderator for fielding all the questions. So we've got a lot of questions. If people have others, feel free to keep adding them into the Q&A. So I'm going to start with Jane. And there were a couple of questions from your talk. And one question was, do you anticipate that there'll be challenges in recruiting people for studies of antimicrobials if people generally are feeling better?
So thanks, Edith, and thanks for the session. It was great and really varied and interesting. I think there are likely to be challenges in recruiting people. I think it's going to be difficult across the board for all types of interventions. People will be feeling better. We're already seeing people embracing more fully aspects of their lives that they might never have thought that they were going to perhaps be able to, and wanting to spend less time in hospital. And the pandemic has catalyzed that very much. So we're now conducting such a lot of clinical care remotely and in the home, that people are not necessarily used to even being around the clinical space very often. So yes, I do think it's going to be an issue. We've been very, very lucky for decades in how altruistic the CF patient and family populations are. And I know that quite often this is driven by a wish to help others and not necessarily themselves. So I think, we really need to continue to work with the community and to try and design trials that they find acceptable and that they can help us actually translate into new drugs, because I strongly feel they're going to be needed. But yes, I think it is likely to be much more challenging.
Dara, I think I'll pass the same question to you. I'm just wondering about if you think there'll be difficulty getting industry involved if there's perceived to be less infection.
Yeah, I think the message is going to be that infections are not going away. Pulmonary exacerbations are going to look different. Sputum may be a little bit more difficult to get. But I think that we're going to continue to try to attract that new research, develop novel methods to really fight infections for people with CF.
Great. Jane, another question, this is an interesting question. With the data following ivacaftor of Pseudomonas burden going down, but then potentially having some rebound, do you think that there are going to be changes in how this presents clinically? I think the question is, do we think that this is associated in the same way with clinical disease as it is in people who are not on modulators, or is there some degree of carriage of this bacteria that may just persist?
Yeah, I think that's a really difficult question. It's very intriguing, and I suspect it's different at different stages of disease. So one of the reasons I think that infection is going to persist is that many of our patients with CF have only got access to modulators since they've already had very established bronchiectasis and quite a significant degree of lung damage. And those people will probably respond differently to having their CFTR function restored than somebody who's actually been able to access drugs at a time when their lungs are very, very healthy. So I think it's likely that the infection course in those groups of patients will be different also. And we may also not see completely similar responses to Trikafta or Kaftrio as we did for ivacaftor. So I think we really, really need to get the data. And one of the challenges, as several of the speakers today have alluded to, is making sure that the data we think we're getting is accurate. And in the absence of sputum, that is a real challenge, because sometimes the decrease in perceived prevalence may just be that the sampling is not as good as it was prior.
Yeah, I think that question came up in a lot of the questions for many speakers about getting airway samples. One interesting question, I think this is also for you, Jane, is whether you're having difficulty with getting sputum in terms of looking at whether a candidate antimicrobial could be inactivated by CF sputum. So Luke pointed out that that's often requested by investigators or companies as part of their preclinical work. Are you seeing that as a challenge to some of these trials?
Well, the first thing I'd say is I think it's a great question, and I think it's totally essential, because I think that people who develop a really promising antimicrobial in the lab and then expect it magically to work in a CF lung are probably missing a number of tricks. So I think it's great if people are thinking that they need to make sure that their antimicrobial is not adversely impacted either by degradation or by binding. And I would strongly support that. And certainly if we're getting requests for advice, we would always talk about that being an issue. Undoubtedly, getting access to that type of sputum is going to be more of a challenge than it was in the past. But there are still patients who, despite being on modulator therapy, are chronically productive with sputum. And my vision might be that we would work quite closely with certain of those patients to use a resource that they're otherwise just going to flush down the toilet or throw into the bin. And actually talking to patients like that, they really would love to be able to help by providing sputum samples. It's just going to take a bit more imagination and creativity, I think.
Just to follow on to that, do you think there's going to be differences in sputum from people who are on modulator versus not, like just the actual sputum characteristics, and will you need to test both?
I think there probably is going to be a difference, because I would imagine that some of the hyperviscosity and the very high solid contents and the high levels of DNA that we see in conventional CF sputum may be quite different in people who have more modulated sputum production. Whether or not we need to test both, I don't know. That's a question I haven't thought about until now. I suspect if this new compound survives the sort of classical CF sputum, then it can probably survive anything. And then maybe that's the place to start.
Yeah, great. Dara, there's a question about sort of how you see the Infection Research Initiative evolving and going forward in the future.
Yeah, so the initiative kicked off at the end of 2018 at NACFC, and we have really made great progress in terms of our funding levels and really attracting some of that new research. So eventually, when the 5 years initiative is up, we need to transition to a more long-standing program. We're starting to think about that now, and we've done some surveying work with the community and researchers to help understand what that long-standing program would look like. Infection, like I said, is not going away, and it's going to maintain as a high priority to the CF Foundation. It's just a matter of how do we continue to evolve as infections continue to evolve.
Great. Yeah. Luke, I'm going to pass the question about sputum off to you and get your thoughts. The question specifically was, how do you see your lab or similar microbiology labs pivoting if sputum is less available?
Good question. And I'm not sure I have a definitive answer, but I think the best answer to that question right now is that we are thinking about it very heavily, and we're talking amongst ourselves. And it's, as Dara kindly and wisely and thoroughly pointed out, it's the topic of some formal discussions, at least within the CF Foundation, and some informal ones I know we're having with our colleagues in other countries. For example, there's this diagnosis and detection, which we all refer to as the Dnd Effort Workshop. You can imagine what the logo looks like, in which a good group of investigators, a really diverse group of investigators, and as well as some industry folks and CF Foundation folks got together to discuss what the implications of changes, first of all, what the changes in expectoration are, and the implications of those changes are, both for clinical care as well as research and the development of new pharmaceuticals. And that's an effort in progress. I would say the main outcome from that, and this may disappoint people, is going to be identifying areas for important research going forward, but also pointing out where we're going to get that information. I don't think it's going to result in a list of recommendations for companies or investigators or even for clinical care at this point. That I think is just going to evolve. The other effort, I'm very fortunate to be involved with Dr. Seaman and John LaPuma and others with the foundation to re-examine our recommendations, the recommendations of the foundation regarding infection control as well as infection diagnosis for clinical care. And that, again, is an effort that's underway. A lot of this is going to be informed by sort of the evolution of, the evolution of clinical care and the evolution of disease in the post-modulator era, with a very close eye to how things are evolving for those who are not yet on modulators. So I think that's a long way of saying we're thinking about it. We don't yet have an answer. But we're talking about it, and it's a topic of sort of intense discussion right now. And watch this space.
Great.
Lisa, is there anything you would add to that, because you're part of all of this too?
No, I think it's really an apt way to transition into that, and I'm looking forward to that also being an international collaboration.
Luke, I have a practical question for you. How would an investigator who's interested in working with the lab, if they're doing investigator-initiated studies or single center, are they able to work with you, and how do they do that?
They absolutely are. The CF Foundation has a very nicely designed website for the National Resource Centers, the NRCs, and the two resource centers that we run here in Seattle, the Center for CF Microbiology and the Microbiological Outcomes Advancement Core, have clickable links there. we frequently provide services to single center studies and individual investigators, and we also refer all of the NRCs refer investigators to the other NRCs as appropriate. So for example, I frequently get requests that I refer to the Microbiome Core or to Dr. Lapuma's Brookfield Area Repository if they have requests that are not appropriate for us. So that's a perfectly good entry point. And we get very frequently get requests for bacterial isolates from archives, for example, from individual investigators, as well as requests for very simple clinical laboratory services, all of which you're perfectly, we would love to hear from you.
Great, thanks. Lisa, I'm going to ask you a couple questions. So I think that viral data, there's a lot of comments on that. Is so interesting. And I think two questions about how you sort of see the next winter coming on. And do you think that the SARS-CoV-2 will, how do you think it's going to interact with other viruses? And then just a follow on to that, like what's up with flu? Like why is that one not coming up?
Yeah, these are the $1 million questions. I feel like answering it the way Jane answered the question about what's going to happen with Pseudomonas. So, I think the one way to frame this also is like when we think about CF in general, we haven't really thought about the impact of the community, and viruses are quite different because the viruses are out there circulating in the community. And just thinking about what we saw and others saw for RSV, it was really different. We're also familiar with the young, otherwise healthy child with bronchiolitis that comes in young, 6 months of age or younger, and needs sometimes to have a lot of respiratory support for 2 to 4 days. This year, with the interseasonality of RSV, we were seeing older children coming in, some of whom had some comorbidities. Interestingly, some of whom had higher BMIs than would be appropriate for age, and really getting sick from RSV. And so the issue becomes how much sort of boosting of immunity happens even with older children when they're in preschool or in elementary school, and how many of those children's older siblings are coming home and boosting their natural immunity. So I thought that's been very, very provocative. And the data that I presented ended in sort of May and June, but if you continue it out through the summer months and into now, the RSV curve looks really, really different. For influenza, we are really just beginning the season, as everybody knows. We recommend that people get generally vaccinated by the end of October, beginning of November. We are starting to hear about small outbreaks, double digits occurring. And we and others are certainly starting to see 1 or 2 cases, and they don't appear to be false positives. These people are truly ill. So I am worried. We know that traditionally the H3N2 part of the vaccines actually have the lowest vaccine effectiveness in general. That tends to have more of those drifty mutations that lead to lower vaccine efficacy. But knowing what we learned from Australia, albeit super small numbers, I think that has to be really, really attended to. And just getting vaccinated is going to be hugely, hugely important. The interesting thing about COVID and co-infection was that during the height of the pandemic in a lot of places, we actually had to divert our resources for the full, what we would call in our center, like the biofilm assay, which a lot of us probably use, these multiplex PCR systems that detect 19 or 20 targets, because we were diverting all of our resources to SARS-CoV-2 testing. So for us, at least, during the really largest surges that we've had, we were really limiting the amount of being able to use the multiplex PCR. We have reinstituted it, but the case counts are much, much lower here. So it'll be very, very interesting because now many of those multiplex systems have SARS-CoV-2 incorporated in them. So we might get a much, much better look at what I think you're alluding to, Edith, is co-infection and the impact. So I'll borrow another, I think we have to just wait and see. But I think that the non-pharmacologic interventions, particularly for mask use in indoor settings are going to hold us in good stead for these other pathogens as well. So I'm advocating for that.
Yeah, that's great. And yeah, I think that's a really good point about the testing being done, that it was really so SARS-CoV-2 focused. And I know our institute has just moved to more the multiplex, or back to that.
It's a really lost, you know, we think back on that. And I remember colleagues saying, can't we save all the swabs? And we were like, the lab just had no capacity to do that.
Yeah, yeah. It's interesting. So definitely some comments about your thoughts about mask use. So I know, do you want to give some thoughts about that you've had?
So mask use is it's been very politicized. And I know that the audience is basically from all over the world. And we were talking before we came on, while we were all in the green room, and it doesn't matter if you're in the US or Canada or parts of Europe, there will be places that don't believe in masks, but the data supporting the effectiveness at masks as both source control, so the individual that's sick, not dispersing as many infectious droplets, and the person who's susceptible, not becoming infected, are really solid. And I don't think that anybody would question that science. We in CF, we've advocated for mask use for our patients around one another. And we have not advocated for healthcare workers, this is all pre-COVID, wearing masks, but just donning gowns and gloves. What I think we need to think about as a community is whether or not we think that we should continue to advocate for mask use for people with CF in either selected circumstances or people with CF who have more severe disease. So I mentioned a couple of things, and this was really, to be honest, more of a provocative thing, like we should be thinking about this as a community. So for example, somebody that has more advanced lung disease, I think that we all recognize that transplant recipients are really, truly different. The data support that people who've had transplants don't make as robust a response, for example, to SARS-CoV-2, and we've known that for a long time for other vaccines. That's the reason why we advocate for a third dose for people who are immunocompromised. And then, of course, they can get a booster dose later. So that's a different terminology. And so those individuals could really be self-advocates for protection against viruses. And people who aren't getting the benefit yet of modulators. And other thoughts about that, maybe somebody that's really a frequent exacerbator, trying to keep them out of the hospital, as Jane said. So yeah, I think it's going to be really, really interesting to see what happens, particularly as this winter progresses, because we know when people come indoors, that's when we start to see all of the viruses start to circulate. And it's getting colder in most parts of the Northern Hemisphere.
Yeah. For other clinicians, like, do you guys, has this changed your recommendations as far as daycare and schools and those kinds of exposures in your younger kids?
Edith, if I can keep the talking there for just one more second.
Yeah,
so this sorry, but the CF Foundation has assembled a group of us to really be their task force for COVID. And I and Patrick Flume serve as the co-chairs of that committee. And, this is just another example of the foundation being really proactive about helping community. And I think that one of the really important things that has come out since, the schools in the US started opening was just how important it is to resume normal life as much as possible for children. That the scars of the pandemic and the social isolation, the lack of physical activity, for some children, the loss of actual academic prowess because of the disruptions were just huge. And so I think that, there's been and if people want, it's all on the website, of course, and people can read more about it. But I think that we have to think about that as well as the concerns we have that people may get a viral illness or.
Yeah, no, I think that's a good point, especially as people are getting healthier and feeling healthier. You want that balance may shift. But.
and now they can get vaccinated.
Yeah, yes, and now they can get vaccinated, yes. Yeah, I don't know, does any, Luke, Jane, Ranjini?
Well, I'm really, really glad that Lisa mentioned that because we're in a sort of respiratory healthcare themed discussion here. I feared that that was going to get overlooked. But certainly in pediatrics over here, we have an absolute epidemic of mental health problems. And it has really been triggered and catalyzed and made so much worse by everything that young people, particularly adolescents, I think have gone through during the last 18 months. And so I've been a very strong advocate of trying to get them back into schools as safely as possible. And certainly based on quite a lot of the evidence that I think I've seen, the ventilation within a classroom seems to be really extremely important, and perhaps more so than masks. Our feeling was after the first wave of the pandemic that the CF children had really fared very, very well from any of them who got the infection. That number was smaller than we might have thought, but actually most of them, at least in the UK and in our clinics, were being picked up because they were getting tested for some other reason rather than because they were really, really sick with the virus. And so we felt that, I think for many of those, that the benefits of so-called shielding were really not commensurate with what they were missing out on. So I think everything does really, really need a very measured approach. And of course, in the UK, we don't have the same approach in general, or we didn't have the same approach towards masking as the US centres did prior to the pandemic. I suspect that there will be probably some continuation of that thereafter. But there is also quite a loud voice from a lot of our CF community about the stigma associated with masking up. Perhaps not so much in a hospital setting, but certainly outside and beyond the pandemic setting, whether or not that will be something that is considered acceptable. I really don't know.
Yeah, that's great. That actually kind of leads into another question for Lisa. The question is, do you think that the CF lung sees different cell tropism for certain viruses compared to non-CF? Can that explain some of what Jane was talking about?
Oh, I don't know. I know that there was some thoughts about that, some of which were debunked over time. I think partially because there was initially people were missing that a lot of the staying home, social distancing, avoiding crowds, school closures was really impacting rates in a very, we didn't understand that fully. I don't know. I'd love to hear from Luke and Jane if they have any thoughts about that. I think it's a very provocative question.
Yeah, Luke, do you want to?
This is outside my area of expertise, but I can voice some of the theories that I've heard kicked around. Just as Jane very eloquently said, I think many of us were surprised and relieved early in the pandemic to find that not only were children with cystic fibrosis faring relatively well, and it couldn't entirely be attributable just to social distancing and those kinds of protection techniques, but people with CF weren't having as difficult a time with COVID infection as we expected, based on what our assumptions that this is a coronavirus, and we've seen that coronaviruses can be airway tropic and airway pathogenic in the past. But that's not true for plenty of other viruses, of course. We've heard, Lisa just told us about influenza and RSV, et cetera. And so when it comes to COVID, Edith, I'm so sorry. Remind me, I'm going back to the daycare question, but tell me again what the question was. So
the question was about whether comparing CF and non-CF.
Oh, tropism.
And right. Actually, they had said, and children and adults, that difference there. Is there differences in how the virus acts with lung cells?
Right. What I was going to say is that with COVID, I don't think we really know. I mean, one of the theories that I've heard kicked around is that this is simply not pathogenic for airways, and therefore, in the way that a lot of other respiratory viruses are. Thanks for reminding me what the question was. In the same way, for example, as Rhino virus or influenza virus, which are absolutely triggers for worsening and acute symptoms in people with a diverse range of airway diseases, chronic airways diseases like cystic fibrosis and other failures or impairments of airway clearance, asthma, chronic bronchitis. These are airways diseases where those kinds of viruses clearly lead to exacerbations. And I think people are starting to wonder whether that is the case for this particular coronavirus. And if it truly isn't as pathogenic for airways, or that's not the locus of pathogenesis, then we're not going to learn much about the effects of this virus, or from this experience on tropism from people with CF versus not CF. But I do think that the fact that as we've heard many times during this conference, that viruses are a very frequent trigger of respiratory exacerbations. It's hard to imagine that there's a lower tropism for viruses in people with CF in general, respiratory viruses. Greater tropism or greater susceptibility. I think it's hard when it comes to viruses. I think it's very hard, and I'll look at Lisa as I say this, to tease apart infection from symptomatology when it comes to viruses in particular. Whether somebody is more susceptible to infection does not necessarily translate into whether somebody is more susceptible to an exacerbation or severe symptoms with that virus. And so I don't know that we can answer the tropism question so much, or susceptibility question so much, as we can answer whether somebody is vulnerable to the symptoms of a particular virus, which I would say yes for everything but COVID, but boy, am I surprised about the SARS-CoV-2 experience. But again, I'm looking at Lisa to see if she shakes her head at me.
Yeah.
I was thinking Jane should weigh in on that question.
Comment?
Well, I mean, it's been a really interesting one, and it did interest me a lot in the sort of early phases of the pandemic. We were all, of course, as many of you were, redeployed onto adult intensive care for a while, and so the sort of impact of that very much seemed to strike me that actually it was almost the systemic risk factors which seem to place people really at risk. So age, metabolic syndrome associated with obesity, diabetes, especially poorly controlled diabetes, and across the board, that people with the isolated airways diseases without those other complications seem to be less at risk. But one of the interesting concepts I've heard voiced about CF people is not, I haven't seen any evidence that there's a difference in tropism for airway cells and the virus, and that seems to me highly unlikely. But one of the things that has been mooted, in addition to their very, very good pre-existing skills at social distancing, is the drugs that they're on. And for example, very severe COVID-related ARDS is manifest by neutrophilic extracellular tract formation. And of course, people are all on DNAs, or many of them are on DNAs, which might mitigate that. There's some concept about azithromycin and anti-inflammatory benefits. So I think that it is possible that it's something to do with, in addition to their lifestyle, the drug cocktails that many of them are on. But yeah, I think it seems unlikely from what I am not a virologist or a cell biologist, but it seems unlikely that it's a tropism issue, I think.
Great, thank you.
I could just jump in, Edith. Really, that's what that SARS RFA was about in 2020, is really trying to attract that research to help us understand on the cellular level how this infection may affect people with CF differently than non-CF. And they are, there were some topics brought up that we're looking at how the medications people with CF take on a regular basis, how that may impact the virus infection. But again, this was all on a cellular level, not a clinical research study stuff. Yeah,
that's great, Dara. So hopefully we'll learn some things in the future. So that's
next year.
Next year. Yeah. Yes. Okay. Actually, Dara, I'll stick with you. There was a question about the remote monitoring, and the question was about recruiting participants. But I think maybe like that's sort of a step ahead from where you guys are. Do you want to talk a little bit about the remote monitoring?
Yeah, so we actually started a remote endpoints task force. It's going to actually look at multiple endpoints, not just collecting sputum at home, but other remote endpoints that we can collect for clinical research. That has really just started. We had a call for people to join the task force. So we really finally just brought the group together. We're going to kick off and really just starting to think about what is out there right now. What are people doing in terms of remote collection? And then trying to understand where are our gaps in knowledge and how do we address those gaps in knowledge. There's been a lot of creativity, though, with our researchers in terms of pivoting to home collection, especially with the coronavirus and labs being shut down and hospitals not being able to do clinical research inpatient. So it's been an exciting time, I think, of innovation on how to get those sputums from home and get those samples from home. So you'll definitely see more of that work in the future.
That's great. Thank you. Jane, there was a question about antibiotics and sort of what is your, if you had to sort of look ahead, is there one group of therapeutics that you talked about that you think is has the most promise for people with CF?
I don't think actually there probably is. I think what we need to bear in mind is that we need to add to the toolbox, and we don't necessarily just need one thing. That it's very unlikely one thing is going to hit it all. But I think progressing the various different strategies, and some of which will work in combination best with each other, perhaps, is a good way forward. And I was very taken by the plenary that's just happened and the concept of different types of approaches coming together to add more than the sum of the various parts. And I think perhaps with antimicrobials, a similar thing might be the case.
Yeah, that's great. Do you think that as people are getting onto modulators and maybe mucus is changing in the lungs, that the tendency to use the higher doses, especially inhaled antibiotics, do you think that that will change, that we'll be able to use some lower doses?
I suppose it might. I suspect that in people with advanced lung disease, quite a lot of the impact of inhaled antibiotics is getting lost because of mucus plugging and the inability to access. Teasing that out is going to be a really difficult one, though, isn't it? I mean, when we think about the history of how topically and inhaled antibiotics has been developed, it hasn't been the sort of field that has gone in for an awful lot of dose ranging. And it's actually quite difficult to do the PK and PD consequences of inhaled antibiotics, isn't it? So I haven't thought about how we might begin to answer that. And given I would say that most inhaled antibiotics are really pretty well tolerated, maybe we should celebrate the better impact of what we're using the current doses rather than try and necessarily come down in them.
Yeah, I think it'll be interesting to see if, does eradication get better? Do we eradicate more? I think those studies might have to be kind of redone in the modulator population. So very interesting. Okay. Lisa, do you want to comment at all on the masking in the non-CF population? Do you think that that's something that's going to continue?
It's very, I think about it a lot, because some of it for me is ultimately going to come down to what does a local jurisdiction mandate. I live in New York. I think most people know that. And to go to a restaurant or the theater, you have to be wearing a mask, show your vaccine card, and show your identification card. So that's just the way it is. But I also think about what people's personal perception of risk ultimately is and what their own risk factors are. So when we get on the other side of the pandemic, and I think that one of the $1 million questions that might have been circulating earlier was, what do we think is going to happen to SARS-CoV-2? Right. Do we think it's going to become like influenza, that we see it every winter? A lot of the coronaviruses, the human coronaviruses, appear in seasonality, and sometimes they'll go away for a full year and then return the following year. Is that likely? There's something unbelievably different about this virus in terms of its success at mutating. but that has to do with the huge number of cases. So as we vaccinate more and more of the population, will we be able to really turn it into a series of small, smoldering outbreaks that we can quickly squelch? The other thing that we have not been able to yet actualize for this virus is a really good oral antiviral, because if we really could manage the relatively less ill patients, that would be fantastic. We would prevent hospitalizations, we would be able to prevent transmission. And it's just, I know people are actively working on that now. So I think that I worry that, although I mentioned this earlier, that we know that masks really do work, that it's become so politicized in certain parts of our country that, and the world, that this will really vary by jurisdiction. And I was very struck by something that I think Jane said this earlier, the concept of people wearing a mask being, and again, this is pre-COVID, it having a stigma attached to it. I'd love to see a time period where wearing a mask does not have a stigma attached to it, that if I choose to wear a mask, then I'm respected for that choice. And I hope we get to a place in the future where we don't need to wear masks, but if we do, I hope that people will be truly respected and honored. And for example, if somebody wears a mask, you know nothing about their personal health history, and they may be just, they are choosing that because of their own risks and their perception of risk. So yeah, long-winded answer, Edith, but I think about this a great deal.
Yeah, no, that's great. There's a question about vaccine. Do you know anything about the efficacy of the COVID vaccine in the post-transplant population?
You know, it's very unfortunate that most of the data in the post-transplant population is looking at antibody levels. And antibody levels are, of course, thought to be a surrogate for protection. But even if you look at the CDC data around the data that suggested the need for the third dose, it was all based on antibody levels. It was actually not based on breakthrough disease. But I think that we can assume that transplant recipients are not going to make as good an antibody response and going to be more vulnerable. The other thing that we have to also remind ourselves is when we measure protection, we're looking at antibody production, but we're not looking at the cascade of T cells talking to B cells. And there's many transplant recipients, of course, don't have as good T cell function too. So that is unfortunate. The great news is, though, you know, monoclonal antibodies for people who are eligible for them really do seem to be very, very effective in the transplant population, as well as obviously other high-risk patients. So I think of that as a really important tool in our toolbox, as long as the variants we have, you know, remain susceptible to the monoclonals.
Yeah, no, that's good. I didn't know that. So that's great. Let's see, I've got, Luca, a couple questions for you. One is there a national resource core that focuses on viruses? I'll ask that one first.
No, the short answer is no, but there is going to have to be. And I will tell you, I can say that several cores have already been either working independently or been asked to work on identifying, titering, measuring susceptibilities to phage. There are one of the efforts of our outcomes advancement core, the basic laboratory, and I think Jane alluded to similar efforts across the pond, is to try to understand what the endogenous, the naturally occurring phage population or virus population in the CF airway, or at least in CF airway secretions, are, how those change with antibiotics, how they change with changes in bacterial complement, how stable they are, so that we can better understand what the landscape is that phages are being added into. But no, there is no, at least there's no core that's specifically dedicated to that. And I do think, I mean, the foundation has clearly shown how interested they are in keeping up with the time. So that's definitely something to think about. That said, again, I think several of us, there are many cores now involved in clinical studies of phage treatment. And so the expertise is being, it exists and is being applied.
Second question for you, Luke, what is the capacity for the core lab in terms of working with international studies or samples?
Right. Very good question. We two answers to that. The first answer is that for standard CF clinical microbiology, we are it's acceptable. We only consider samples that have arrived within 48 hours of collection acceptable for standard culture-based microbiology. For molecular biology, for qPCR-based or other DNA-based or sequencing-based or microbiome-based outcomes, we can accept frozen samples from whenever, collected whenever. And so in general, it's very, very hard to get samples that were freshly collected samples to our laboratory within 48 hours. And so for that reason, answer number two, we have partnered with other laboratories in other countries to act as basically to mirror the methods that we use to make sure we harmonize to serve multinational studies. We have we have a laboratory in the UK who we've worked with before, and a laboratory in Australia who we've worked with before as well. But again, I think our hope is to generate a more robust and formal international relationship between these core laboratories for exactly this reason. We have, however, again, we've done molecular testing, looked at molecular outcomes for samples from other countries in a number of studies.
Great. Okay. Well, I think that we've hit most of our questions. So I think I may just go around to speakers if you guys have last thoughts about this. Dara, you're on my screen.
Yeah, I just want to thank everybody, especially everybody on the screen, just for being incredible researchers and helping people with CF. Your time and dedication is always appreciated. So thank you.
Great. Well, I'll just speak for us that we thank you to the Foundation for all your support of these studies. And Luke.
I'm sorry, I was answering something.
Oh, sorry.
I'm not doing a great job right now. Thank you very much to the organizers for inviting me. This was a great discussion. Thanks to everybody for attending and for their questions.
Great. Lisa?
I just want to tell everybody that to get vaccinated and to get their 5 to 11-year-old patients get vaccinated. I know there's a lot of work being done by all of you. There's a lot of vaccine hesitancy out there, but data suggests that the people that patients and families want to speak to are trusted providers. So you are them.
Great. I'll just give a call. You had done with a group of people, but Lisa, you had spoken in a seminar about talking about vaccine hesitancy. And some of those talking points have been so useful in clinic. So if people haven't seen it, I think it's still available on the Foundation. But it actually gave me some very clear messaging. So very helpful. And Jane, do you want to?
Yeah, well, just finally, I'd say, it's fantastic the progress that has been made with the highly effective modulators, but I really don't think that it's time for us to go and push our feet up about infection. I think we're going to have an infection problem for the foreseeable future. So I'm really pleased that this session happened, and I hope that people out there who are thinking of working in this space, either academic or clinician or else industry, really feel fully sort of motivated to carry on with it, because I really think we've got a lot more work to do. Thanks.
Great. And Ranjini, do you want to comment? You've been doing the hard work of all the questions.
Not at all. Thank you again, everyone. This has been such a great session and great to be part of. And I very much concur with Dr. Davies that I think there's lots to be done in the infection space, and I'm happy to be part of this space.
Okay, well, thank you again to all of our speakers.
Bye.
Bye.